Welcome to All the Best, an interview series with UNL Orca awardees, where UNL means University of Nebraska-Lincoln, and Orca is the Outstanding Research and Creativities Award, which is UNL's top award. In Gerda Lerner's book, Why History Matters, she explains the life stories give meaning to the present and the future because we identify what is important to us. And she says, our stories show how we are interconnected in a particular time and place. As for this interview series, Orca awardees have experiences that reach back more than a half a century, which will give us a personal view of how the best research emerged from UNL. It is my pleasure to introduce today's interviewee, Dr. Stephen Baenziger. He was awarded the Orca in 2011. After I provide a quick summary of his life and career, Stephen will tell us about his time at UNL, starting with his award-winning research. Stephen was born in 1951 in Toronto, Canada. When he was an infant, his family immigrated to the US, where he grew up in Des Plaines, Illinois. He earned a bachelor's in biological chemistry, magna cum laude from Harvard University in Cambridge, Massachusetts. Next, he earned a master's and doctorate in plant breeding and genetics at Purdue University in West Lafayette, Indiana. Over the next decade, he worked for the USDA at Beltsville Agricultural Research Center in Maryland, and then at the Monsanto Corporation in St. Louis, Missouri. In 1986, he joined the University of Nebraska-Lincoln as an associate professor of agronomy, later agronomy and horticulture, and rose quickly through the ranks while studying the genetics and breeding of small grains such as wheat, barley, and triticale. He retired in 2021 and still lives in Lincoln. With that, I want to welcome Stephen to the interview.
Well, thank you for having me. It's a great pleasure to be here.
I'm excited to hear this one. You're our first agronomist on this podcast. So my first question is always, what was your impression about the UNL campus when you saw it for the very first time? And what do you think about it today?
Well, I always laugh because I came in a heat wave. So I was here in March, March. And I think the temperature was 60 degrees.
Oh,
And so I was teasing my department head who was squiring me around if this was a normal March because I've lived in St. Louis and we would expect much colder that time of year. And I was applying for a winter small grains breeding program. So weather is very important. So that was my first impression. My impression now is I've always loved the landscaping, and the university has done some remarkable things there. And then if you go to the East Campus Union, they've improved it and added on and things. But functionally, the parts they didn't change is almost the same as what it was when I was there in 1986, 40 years ago. It's amazing how little it's aged.
do they always have the bowling center?
They always have the what?
The bowling center?
I believe they did. Yeah. Yeah. No, I think that was there when I was there.
Okay. Yeah. All right. So let's jump right into your award-winning research. Give us a sense of what you think you won it for, and then why it's important.
Okay. So the research, and I'm probably an outlier compared to most of your interviewees, because I actually created new varieties. Mm-hmm. And so I won it in part for the varieties I developed. And our tagline is, if you eat bread, you eat our wheat. Mm-hmm. Because at one time, 80% of every wheat that was grown in the state came from the Nebraska program.
Wow.
And that would extend, of course, beyond our state borders. Wheat doesn't know a state border. So I won it in part for that because we had tremendous impact. And then secondly, we won it because of the research we did on how to do better breeding. And so we did two ways. And there's an old story of one of the Persian kings who asked an architect to give him a plan for a bridge. And the architect built the bridge. And when the king came back, he beheaded him. And he said, look, I could have anybody build the bridge, but I need to have plans that I can send over my, at that time, sprawling empire, so that anybody could build a bridge. And so that's kind of where a plant breeder is. You have your impact, but you also have to document how you do it. And so I probably won it for those two things. Okay. And we did a lot of things that it's easy for plant breeders to let the inertia of the program, you certainly do certain things, but you should be curious about every step. And so we did a lot of research that was curiosity driven, which is somewhat unusual for applied scientists, but that's what we did. And so that's what we went in for. And we have a number of things that we were very much involved with.
Great. So you were at the university for 20 years before you got it, roughly?
Well, I was at the university from 86 to 2021, so that would be 35 years.
Okay.
And it was 20 years, you're right, before I won the award.
Yeah. So that's a lot of summers and winters to grow wheat and optimize each step, or what really is the goal?
Well, the goal is to provide the grower with the very best variety. And we had 4 characteristics it had to have. It had to survive the winter. That's pretty obvious. It had to have good yield because that's what they get paid for. It had to make a loaf of bread because that's how it's used. And it had to have stem rust resistance because when that disease came, it comes like a sign and it's absolutely devastating. And the first year I was here, and my predecessor, John Schmidt, who was one of the great breeders of all time, he made a decision in the 50s, because we had major stem rust epidemics, that he would never release a variety without stem rust resistance. And we kept that for my tenure, because the first year I came here, his wheat went 55 bushels with about a 59 pound test weight. And a susceptible line went 17 bushels and it had a 42 pound test weight. So they lost 2/3 of the yield and then they lost at least 1/3 of the value just due to worm disease. So it was kind of a first impression that lingers with you long. Now, we never had a stem rust epidemic in the next years that I was here or for the next 40 years, but it always could come. And that's what the question is. You don't want to be unprepared.
So how do you breed wheat?
Well, okay, so
it's all down to the seeds, but what do you do to the seeds?
Well, so the traditional way of breeding wheat is you make a cross, okay, and then you allow the variation in that cross to segregate. And wheat's a naturally inbreeding species, as is barley and triticale. So then you allow the segregation to separate out the variation you put in, and then you select the best. And then you evaluate to make sure that they're still good. So if you want to know what makes a plant breeder happy, it's when they first decide what cross they're going to make, because that's when you think of all the good things you could get. The second time is when you're out in the field and you see a selection which is going to make it. You can tell that it's got, it has the appearance of a new variety. And then the last thing is when that variety is grown on 1 million acres. And you remember when you held every seed of it in the palm of your hand, which you can do. And so that's sort of the impact that plant breeding has. I mean, it's something that's very tangible, very visual.
So you're breeding these plants and you're growing them on a small scale, but at some point you're going to have to make a lot of the seeds to distribute.
Right.
How does that work?
Well, you have very set organizations that help you along the way. Okay. So normally we would get to roughly 60 pounds of seed.
Okay.
We would give that to the foundation seed organization. They would blow it up to 400 bushels, which would be, that time 400 would be, I was going to make sure I got the numbers right. 2,400 pounds of seed, 24,000 pounds of seed. That would be sold to the certified seed growers. They would multiply it. And to plant 1 million acres, you would need 60 million pounds of seed. So that's the way it goes. It just goes year after year.
Yeah, so that's like 4 years of prep before
it's 4 years of increase before it gets too large. Yeah, historically, you could do 1 year to get from 60 to 400. No, 60 to probably 40 bushel. And then after that, you would get 400 to 1,600 bushel. That's when they would sell it. Usually they said you would need at least 2 years at the foundation seed, 1 year in the certified seed, because if you sold 1,600 bushel, you could get from that 64,000 bushels. That would be the initial launch. So it'd take 3 years if everything goes well. And you don't wait. What you do is you sort of plan that one of these will work. So you give them like 4 varieties. Well, no, and you're not sure which. And then they look at them and you look at them and you decide which one will make it. And then you increase that one. And then you release it. And all the while you're building the test data so the grower knows why they want to grow it. And so you do everything, it's not one after the other, it's concurrently. That's what you have to do it.
And then how much feedback do you collect about weather conditions and agricultural treatment of the wheat? And then how many years after the initial?
Well, it historically would take 10 years from the cross to the release.
Oh, okay.
Usually 12.
Okay.
Okay. And in that time, you would have probably 40 environments. And then when you put it in the state variety trial, you had 15 to 20 every year. You put it in the regional nurseries, which would be another 30. So you'd have well over 100 location years of data. And that would still only be able to tell you what you think you know, because when you put it on somebody's farm, all guesses are off.
Right.
And you are surprised occasionally. We had 2 varieties that were, I thought, excellent and absolutely initially failed. One was a variety called Rawhide, and it was the toughest wheat I've ever seen. That's how it's got its name. We had a drought at North Platte. It went 50 bushel per acre, and the nearest line to it was at 34. So you think, 50 versus 34, that's a tough wheat in a drought year. And the year we released it, we had the warmest end of January in history, or very warm. Anyway, it was 70 degrees in Hays, Kansas on January 31st.
Wow.
Yeah, very unusual.
Wow.
And on February 1st, a cold front came in. The high was 32. On February 2nd, the high was 10. And on February 3rd, the high was 0. And what happened is the wheat had broken dormancy and had massive winter kill.
Oh, wow.
And so that basically, you only get one chance to make a first impression, and that hurt it.
Wow.
We had another wheat called Windstar, which was fabulous wheat because it didn't get blown out of the field. Wind erosion is a problem. And this is a wheat that could hold its ground, but it was the most photoperiod sensitive wheat I've ever seen. And we had a very early year. We normally flower on May 20th. This year we flowered on May 6th, so we were 2 weeks ahead of normal. Because it was waiting for the right day length. That's what photoperiod is. It didn't flower until the 21st or so. It was a relatively late week, probably more like 23rd. And then we were harvesting on June 10th. So it had no time for grain filling at all. And it failed. So you have those, despite 100 environments of testing, it still happens.
And so most of the wheat is grown, Kansas, Western Nebraska, South Dakota, North Dakota, anything in Canada?
Oh, none of ours. That's all spring wheat.
Okay.
Yeah. The spring wheat border used to be midway in South Dakota, but with climate change, it's now all the way up into South Dakota, a little bit more into North Dakota. Our wheats historically have been grown in Nebraska, a little bit in Colorado, a little bit in Wyoming, a little bit in Kansas, basically Nebraska and the adjacent area. It's pretty rare that you get a wheat that goes very far south because the climate's different and it's not bred for that climate.
Too warm.
Yeah.
Okay. And so you're always chasing the future, right? So you got to plan ahead. And what sorts of properties are you typically trying to select for?
Well, that's
those 4 parameters you're trying to optimize.
Yeah, those 4 are what you have to have.
Yeah.
Okay. And the thing you have to understand when it comes to breeding is if you make a cross and 10 years later, 12 years later, you get a variety, you have to think what the trend is going to be in 10 or 12 years. And so in science, that's pretty rare. That's difficult.
That's really hard.
So you watch. things like when there's a new pest, Russian wheat aphid came in my career.
Oh,
Wheat stem sawfly was only a Montana problem, then became a Nebraska, Kansas, Colorado problem. So you have to have diversity so you can adapt. Climate change is, when I first came here, every wheat we released was photoperiod insensitive. Now probably half and half. And that's basically because the photoperiod, I take it back, photoperiod sensitive. That meant that if you had an early frost or wheats were late enough that they could miss that frost and still grow. When you go to photoperiod insensitivity, it's just all done by temperature. You have a warm spring, they fly with that. And then if you get a cold snap, they're caught. That's what happened to rawhide. But so you see those types of things. And so, what most people don't recognize is that a plant breeder should be curious at all times, watchful and curious. Those are the two things.
so pest management, you've talked about diseases, but pests, some are generic, right? Grasshoppers and mice and voles or whatever.
Most of those are minor.
Okay.
Okay. In our, you can have a bad year. The ones you watch for are the widespread ones, which are usually the rusts.
Okay.
Rusts never sleep. And that's why we've always had fantastic plant pathologists to work with. Plant breeding team is not just the breeder, it's the full team. So a good team would have a pathologist. If entomology, insects are important, and it could be things like the Hessian fly, could be like the wheat stem sawfly. It could be viruses that are vectored, such as barley illodwarf is vectored by aphids. Wheat and streak mosaic virus is vectored by an arachnid, the wheat curl mite. I think that's right. So I think you should be careful. Anyway, it's vectored by mites. So you work at that disease, insect, or pest post interaction. And so you have those people involved. You have to have a serial chemist. We've been lucky, Paul Matern and then Dave Shelton and then Devin Rose. All are superb. They all added to that. So you have to have that kind of effort to work with. And I'd be remiss if I didn't say a lot of our research was to do better statistical approaches, which kind of breaks my heart when I see what happened in the statistics department. But I mean, a lot of my papers, probably a quarter to a third have a statistician involved to make sure the statistics are right, because we're looking at very fine differences. And that's really where the difference is. You need good statistics to do it. People who say, if it's not an order of magnitude, I'm not interested, is ridiculous. That would be 100 bushel wheat versus 10. If you can't tell that difference, what good is it?
Right. There's going to be some biological limit of the plant, right? So what about genetics? Did you do any?
We did the genetics, because again, first of all, we're always curious. And in breeding, there's a lot of theories, and the theories need to be proven. And so the first question was, is yield, which is classically considered a quantitative trait, when you measure, you can't find major loci for it. Is that real? And there was a very tremendous set of geneticists here, Rosalind Morris, who created a series of lines where you could basically evaluate chromosome by chromosome. And wheat has 42 chromosome pairs, or 21 chromosome pairs, 42 chromosomes. So you could look at the contribution of each 21. So that's 5% of the genome. Wow. You could do it in replicated trials. She developed these things. And we took her material and did the yield trials. And these were massive. They were, I think we ended up with 8 environments, and you're looking at, multiple lines, multiple reps. I mean, I think our degrees of freedom were over 2,000. So for statistics, we really got in. And we found that one chromosome 3A, from Wichita, when put in Cheyenne, would increase yield by 15%.
Wow.
What was even as interesting is if you took the Cheyenne chromosome and put it into Wichita, you decreased its yield by 15%. So you had a feeling there was a mirror image going on. And this is before there were molecular markers, so you answered it. So you used the technology you had to answer the key question. When we got molecular markers, then we broke the chromosomes apart and we discovered there was a major locus, QTL is what they call it, but a major region that controlled yield at Wichita, and the same region is the one that reduced yield from Cheyenne. And so we started to, that gets at the idea is, can you make the genetics of yield based on Mendelian genetics? And so that was a major part of our work. And it took a lot of time, but it answered a very fundamental question.
So that was in the 90s.
That was in the 90s.
Sounds like it was in the 90s.
Yeah, it was in the 90s. We did that. And then the work on that extended up through the 2000s. Because we got more lines, we got better markers, we got a lot of different things. And the other part that was interesting is, if you think about breeding history, if you have a major gene, you should probably fix it. If you're selecting, you should be selecting for the traits that really have an advantage, right? So we went back and we scanned wheats from Cheyenne, which was released in 1932, Wichita, which was in the 1940s, up to current. And what we found out was every line that had high yield potential had the Wichita lead.
Oh, wow.
Lines that necessarily, because there's certain parts of the state where low yield was not a disadvantage. In fact, the drought tolerance, the stress tolerance, their optimal yield is going to be 30 bushel. The Cheyenne and Wichita alleles don't differ at that level. They only differ when you get to the very high yielding ones. And so some of those varieties that were specifically for the toughness that Cheyenne was bred for, they kept it. And so you start explaining how the pedigrees. And then the other thing we found is that many of the newer lines had neither the Cheyenne or Wichita allele at all. They had something completely different, which meant that we found new variation, put it in, and that was even better.
But there must be many alleles that influence yield.
Oh my gosh, yeah. And in different
Got to be
ways.
thousands.
Well, I wouldn't say there'd be thousands, but there's enough that you can't, it's rare that you can find the one that gives you the 50%.
Sure.
And even that probably only worked because we had these specific lines are called chromosome substitution lines, were so powerful. And I have to admit, it took Rosalind her whole career to develop. And so we kind of came in as she was getting close to ending. And it was like a gift from heaven. And then, the other thing we did in genetics, we did a lot of work on just understanding traits. We dissected the resistance of gauge wheat, which doesn't mean anything to anybody, but it had the best stem rust resistance, most durable stem rust resistance. We looked at that. Because in an earlier program, I had worked on a program where there was a lapse between breeders. So when I came in, there was virtually nothing. Speed meant everything. We did a lot of work with Horticulture and doubled haploidy, and that's now become very popular because time is money and speed is money. And so Horticulture, not Horticulture, but the doubled haploids, which is a way of taking an F1 plant, reducing it to its gamete, doubling the chromosome so it's now homozygous, and then doubling it, and then you can grow that plant, and it's pure breeding. So instead of 12 years to get a new variety out, you get it out in 5. That type of thing. So we did a lot of work in that area too.
So let's go to the next thing here. Tell me about one or two of your early graduate students, postdocs, or visiting professors, and what projects did they do, and where are they now?
Okay, so we've always been lucky. Okay, we've always been able to attract good students, good postdocs, and the like. And so the very first student that I had was the very last student my predecessor had, John Schmidt. And there were two. One is Abdullah Wasu, and he's on what was then the MEAC project. We always had a connection internationally. And Wasu went on to become an excellent professor at one of the major universities in Morocco. He's since retired. Tom Payne was just a wonderful gentleman, and He went on to become the head of the wheat germplasm collections and the international nurseries for Simon. And he was also the secretary of the board of trustees for quite a while. So they've done that. But, every student has its own uniqueness, and every student succeeded in different ways. One of my students went back to Indonesia and ran their international atomic energy effort, which was, most people think of atomic bombs, but it's actually medicine and it's agriculture. He may have been head of the agricultural section. And he was a superb administrator. That was his skill. Other ones went on. I've got a number of students in industry that are applied plant breeders. They've done very well. So they've all done that. The postdocs, generally, it would depend upon what their interest was. I haven't even talked about our genomic selection, but we were one of the ones that, if you look at it, we used chromosome substitution lines because they were available. Then we went to marker-assisted selection because that was available. Then we went to genomic selection as the technology evolved. And we learned an awful lot about how to use that to buttress what we could do phenotypically with our regular breeding program. And they're mainly with companies now working at things like that, or breeders themselves. So many of them came from a quantitative genetics program, learned breeding with us, went back to become breeders, or stayed within the field.
So did they know how to breed plants before they came to work for you, or?
Not necessarily.
Not, okay.
Yeah, everybody that came learned breeding. That was the thing. I wanted every one of my students to be able to breed. That didn't mean they had to breed.
Right.
But I wanted every one of them to be able to breed. And so they did that. And, it's funny. One of my students literally became Dr. Pepper. He became the pepper breeder for Savannas, I believe. And, he won all kinds of blue ribbon awards for his peppers. Another one of mine is a vegetable breeder. Not sure of the crops because it's in Thailand and they have different vegetables.
Sure.
So there's just a lot of people like that. They've had a good time.
Sure. Sounds good. So I'm a biochemist.
Yeah.
Every student did a different project, but every single student isolated proteins.
Yeah.
And what did they get hired for? Because they knew how to isolate proteins.
Yeah.
So it sounds like the same sort of deal.
It is. I mean, there's a lot of people which can specialize, but I wanted them to not only specialize, but to be able to communicate. And in the field I'm in, most of them would end up either at a breeding company, at an academic situation where they would be related to breeders, or at a university. And I wanted them to be able to communicate. I also wanted them to understand. The number one question a plant breeder should really be asking is, when you get a set of data, why? And there's certain things that just happened. Like this year, for example, we had a horrific drought. And if the drought persisted, the early lines probably did better. If you got late rains, the late lines did better. Now, does that mean next year they're going to be the same way? And the answer is no, clearly not. But you need to know that. You need to know which of the lines that are late did well under drought early, which of the lines that were droughted and finished early, so they never got the benefit of the late rains, which of those did they benefit or did well? So it's that constant type of question that you have to watch.
So these, the farmers who grow these plants, how much do they consider all these factors? Do they choose one that's an early?
They always choose one based on what their needs are.
Yeah.
And we try to help them with their needs. So like we recommend to growers that they never would grow all one variety.
Okay.
And the reason for that is genetic vulnerability.
Yeah.
If you have a variety that's susceptible to stem rust, not that ours were, but say leaf rust or stripe rust, you either have to spray with the fungicide or you have to do something else. So you want to do that. We recommend you don't have the same maturity because nobody can cut 1,000 acres in one day. So you want to cut the early ones, then you want to take those same combines to cut the medium ones, then you want to take those combines to cut the late ones. and so you can stagger your workload.
I see.
So there's just a whole series of practical questions that growers should have. So our goal would be to give them the information to allow them to make the decision. They know their farm far better than we
Oh,
do.
yeah.
And so we just have to provide it, and they'll want things like how long is the coleoptile, which for most people don't know, but that's when the stem comes out of the seed. It's the sheath that covers the first part of the stem. And that's important because how deep you plant it. If you plant it too deep and the coleoptile is not very long, it may never emerge. I've got good drills and lots of tricks like that, but then things happen. You plant in a furrow and you plant 1.5 inches deep, and then a windstorm comes up and fills the furrow, and now you're 3.5 inches deep. You better have something that can grow through if that's what you have to worry about.
Right.
So we provide them the information, they'll make the decision.
No, that sounds great. So what was your most unexpected research finding?
My most unexpected research finding. Well, one that we always thought was unique, I guess. Well, there's a couple of things, I'll tell you the one that I think is probably the most unexpected. We were doing a nitrogen use efficiency study with an excellent student, Mary Gutierrez, who's now USDA at Kansas State University. And we did a complete mineral analysis because the question was, is nitrogen use efficiency masked by, say, a mineral deficiency?
Sure. Yeah.
Okay.
Like copper,
Yeah, it could be.
vanadium, or whatever.
Yeah. And she found a line that we had released called Panhandle that was very low in cadmium. Now, cadmium's a toxic heavy metal.
Yeah.
And fortunately, our soils generally don't have it. We found it at Mead, where it was an industrial site for World War II. You had a lot of washing of chemicals,
Yeah.
and the soil was very high in cadmium. We found the first cadmium uptake or reduced cadmium uptake gene in Estevan. It had been known in durum wheat, which is a relative of wheat, for years, but not in Estevan. So that was one surprise. Okay. Yeah. The other surprise was that you could actually, what we would call Mendelize the genetics of yield, those chromosome substitution lines. And then we used a different statistical test, which is called a sign test. Now, a sign test is where if you flip a coin, half should be heads, half should be tails. And the sign test basically tests whether or not that you're getting that 50-50. So we looked at all of the Wichita lines, the substitution lines, and we discovered that even though we could only identify, basically in that case, 2 QTLs, one was, well, there were 3. One was on 3A, one was on 6A, and then one was on another chromosome, but that was a winter hardiness gene. And that was unidirectional. So if you took Cheyenne and put it in Wichita, you had no effect. If you put Wichita into Cheyenne, you had a drastic effect. That was because you took away the winter hardiness of Cheyenne. But when we did the sign test, we found that by and large, in addition to 3A and 6A, the 2 major chromosomes, there were more chromosomes in Cheyenne that had too small for a traditional LSD 5% significant test that contributed to yield. So it kind of came back to that infinitesimal model that it's a lot of little genes making an effect. So there were major genes and there were little genes. And that was to me, it was, I don't want to say it was a surprise because we looked for it.
Right.
But it was interesting to find.
Sure. Yeah, no, statistically that's complicated. Let's see, let's talk about citations. And for this one, what I wanted to talk about first, or have you talk about first, is in your field, everybody gets evaluated every year. You got to submit your findings. How much of a role did citations play in that? But then also, when departments hire new faculty and the candidates don't have much of a track record, how do we pick out people who are going to have a good impact? So blend those two for me.
Yeah, I can. So you kind of hit on on a funny story, at least on part of my career. And I'll give you my background. So when I went to Purdue, I worked on a laboratory master's, and I did that for a solid year. I went there in July, and I worked the summer to learn how to work with corn, solid corn, and worked all that year. And then in the next summer, I started my PhD. And so I was doing a field study. So I'd work all summer on the PhD study, and then when the harvest was done, we go back to the lab. And I worked on that. Good. And then next summer, I'd work on the second year of the PhD study. And then I would work in the lab. Well, so I basically finished my master's in 2 years and was going to, I forgot to apply to graduate in December. So
Having too much fun.
well, yeah, I was probably ignorant. I'd never done it before, so I didn't know you did it. So I actually graduated with my master's 3 years after I started, and I graduated with my PhD 6 months after my master's, which looks a little odd. So you simply say, I got them both in 1975. Okay. But I'm a little, it's funny to me because my major professor was a wonderful man. Okay? But he was very slow on reading publications. So when I went to the USDA, I had, my master's was published by then. And I think I had a very small, quick, easy kind of dirty little publication. That one you would ever cite as your best ever. So I had 2 publications at the end of 3 years. At 2 years and 9 months. And that's when they wanted to put me up for promotion. I said, I don't think that's going to work. And I said, okay, if you want, we can delay it 6 months. So in the next 6 months, more of the PhD papers came out, other papers came out. I had 6 publications.
Oh, wow. That's pretty good.
Well, if we're an agronomist, because time is so important. So I went in late with 6 publications, and I went from what was called the GS11 to a GS12. Mm-hmm. And they almost said that I almost qualified for another grade, GS13. Mm-hmm. And then after that, things just started to roll. And it was always because of collaborators. I've always had fantastic collaborators. Mm-hmm. So my favorite story along the line about citations and what I tell people is do your job. At the University of Nebraska, they only have 1 small grain sprayer. If I'm supposed to release varieties and publish and teach, and I've always taught, and I love teaching, you do it. And things will take care of themselves. Okay. And that's what I try to instill on any young fan, is that you do all aspects of it. And, in my case, I never felt that the varieties I released should carry my academic career. But there are benefits. When you release a variety, that counts the equivalent of a publication. There is a way of writing the variety up as a crop registration. That counts as a publication. So you get the benefit of the university likes the impact. You get the release, you get the registration. So there's kind of 2 publications and impact there. But that shouldn't be enough. You should be teaching. You should be doing, you should be showing the plans, how you do things.
The whole package.
Yeah, it should be.
Yep.
It should be.
Yep, we are at a research university and we do teach, right?
Well, yeah, I mean, I think scholarship and teaching, some of my better papers are what do the graduate students in the future need? That's part of the job.
Yeah.
You should be planting that seed for the next generation.
Right. It's our job to think about the future.
Yeah, and it's our job. And this is something which I had, I think, 40, 50 plus PhD students and another 30 master's students. It was close to 100 students. Now some were masters and PhD. But what surprises me is out of all those students, if you weren't an international student going back to your university, very few of the American students want to be academic.
Right.
It makes me think, what if we taught a student when the only thing they know is an undergraduate, graduate program, and at the end of it, their number one conclusion is that's not for me.
Right.
Now, I'm not saying that's not a bad choice. I mean, that's they're all successful,
Sure.
but it does make me wonder if we've made academia in a way which is different than the way I was in my youth. And that's on us.
Mhm.
You know, we haven't thought about that. We all want prima donnas. We all want, the key ones, the people who win orcas. No, that's not right. You want a lot of people that do the job and do it well. Not everybody can win an orca or an Ortica or any of the other things.
Yeah.
You need good people.
Yeah. No, I agree. Let's look at then some of your papers.
Sure.
The earliest one that I found here, you've got a lot of papers
Yeah.
and very high citation rates. And I picked some that are above 487 citations.
Yep.
But the earliest one on this list is from 1992. It's called Genotype and Environment Effects on Quality Characteristics of Hard Red Winter Wheat. It has 526 citations. Tell me about that one.
Well, so first of all, this is the benefit because I was the third author. Okay. Jim Peterson, who was the first author, was an outstanding USDA scientist. Bob Graybosch, I knew from Monsanto, and then he was hired by the USDA here. He's an excellent biochemist, serial chemist, and geneticist. So this is a great collaboration paper. Okay. And it's just because you have teams. And what we did is we grew varieties in many different environments. And then we wanted to see how does the environment affect the end use quality. So there are lots of papers on how it affects you, but there weren't that many on how it affects that other trait which is so critical for wheat, which is end use quality. And Jim and Bob, and I was fortunate, and Alan Grumbacher was a graduate student. They did an excellent job of really partitioning how much was genetic, how much was environment, how much was the interaction of the two. And so that's what that paper was. And the reason why I get so many citations, the papers that get citations are usually method papers.
Yeah.
The other ones are ones that people need to buttress what they're doing. So the genotype by environmental effects for quality, this is one of the early ones. It was one that really looked at the Great Plains, and it became sort of an early paper that everybody would cite thereafter. And that's why that one was there.
So foundational in the quality. What was the conclusion? Do you remember 50% gene and
Oh, it depends
50%
on
environment?
no, it depends on the trait. That's the key part. So like if you look at something like protein, now you got to remember that wheat's purchased on yield. Okay, that's how the grower makes money. It's also purchased on what we would call test weight or grain volume weight. How does it pack? And that's allegedly related to milling, but not really. Okay. And then it's on protein because you need 12% protein to make a good loaf of bread in a kernel. When you mill it, you need 10.5. So you lose 1% to 1.5% in the milling. And so that becomes huge. And as I recall, protein is relatively low heritability because the environment makes a huge difference. And then you'd have other things like mixing strength, which is how long it takes a mixer to mix the dough. That was higher heritability, which means more genetics too. Okay. Now, again, what people don't recognize is that if you breed to a uniform standard, you often breed to the same genetics. And by doing that, you lose your genetic variation. So the heritability becomes much lower. So, if you want to get high heritable, you take something that's completely disastrous, cross it by something that's extremely good, and then the differences are all genetics and the environment doesn't make as much of a difference. But if you're making good by good, so many of the traits are already identical or fixed, then the environment plays a much higher role. And so this was using the environment and the genotype, very, very elite ones. And that's why it became so important.
And so 92 agriculture in 1992 probably looked a lot more different from what it looks like today.
Actually, not as much as you would think.
Is that right?
I mean, they knew the fertilizer revolution had come, the herbicide revolution had come,
Okay,
all those types of things.
those were what I was thinking about.
Yeah, no, the fertilizer revolution came probably for sure began in the Second World War, but the Green Revolution weeds were all based on fertilizer.
Okay.
So that would be the 70s for sure. And we had history. I mean, if you go to the agronomy department, the history of fertilizer is really well documented for years.
Mm-hmm. No, it's chemical, so I've thought about that. Then the next most recent paper we've got two from 2015. I'll look at this one. Exploiting genetic diversity from land races in wheat breeding for adaptation to climate change. 609 citations. Tell me about that one.
Well, this is a great story. This was Marta Lopez, who was an outstanding breeder for CIMID. Now, CIMID is the International Center. It's in Spanish. That's why it's called CIMID. Centro Internacional de Mejoramiento de Maize y Trigo. But it's actually International Center for the Improvement of Wheat and Maize. She was working in wheat. She was in Turkey. Ibrahim was the second author. He was just a brilliant quantitative geneticist breeder who worked with me. And then the other ones would be the team from Marta's group.
Mm-hmm.
And what they wanted to look at there was in breeding, everything is based on genetic diversity, genetic variation. Okay, because that's what gives you your heritability. That's what gives you your genetic basis. We work on genetics. The environment is what an agronomist might work on. Do you add fertilizer? Do you add water? Do you do these things? And then you have the environment you can't control. But breeders work with the genetics. So the idea is, are you losing traits as you're making improvements? And so it was looking at land races. Now, the land races would be the varieties that were grown by farmers before there were real breeders, you might say. So they would be farmer selections that were shared and grown, became popular. And the idea was looking at those land races to see if they had physiological traits. And that's what Marta was good at. She was a physiologist. I'm a geneticist, so she was the one who was specialized in that, to see if there are traits that really belonged in modern wheats so that we could adapt to a climate change, which we would hope the land races had seen, but perhaps the last 50 years of breeding had. That's why that one became important. And the idea of genetic variation is fundamental to plant breeding.
Sure. So this other one from 2015 is management of Fusarium head blight of wheat and barley. 487 citations. That sounds very important.
Well, okay, so it is, and I want to acknowledge a number of people on this, because if you look at it, the lead author was Stephen Way Gould, and he was the pathologist that I worked with for, well, the whole end of my career. I don't know when Stephen came, but he's an extension specialist as well as a great researcher. John Oaksa was his student. Bill Bacchus was at K-State, and their pathologist. And then Heather Hallam Adams is in food science, who works a lot on mycotoxins, food safety, things like this.
Okay.
That's it. So that one, again, it was just Stephen being very gracious. I mean, I helped him in the field and that kind of thing. I helped with the writing a little bit, but he is the brilliant plant pathologist. And what that one is it was a wonderful review paper of how you can manage a really nasty disease, Fusarium head blight, in wheat and in barley. And so it became widely cited for anyone that wanted to publish in Fusarium head blight, because you say, okay, if you want to know a lot about how it's managed, go see this paper. And that was Stephen's work, and he was absolutely brilliant at it.
Okay, then let's do this one from 2020. GWAS, fast forwarding gene identification and characterization in temperate cereals. Lessons from barley, a review.
Again, your reviews are the ones that get the publication.
Absolutely.
Because basically it describes how breeders would look at it. These are the architectural plans for those bridges for other people to work on.
Right.
And this one, GWAS, is genome-wide association studies. And it becomes a huge part of what later became genomic selection. And it was how we could characterize materials and look at it. And so, just to give you an example, we used to have molecular markers for about 40 important traits. When we do genome by sequencing, we would have all 40 of those traits fall right out from our dense genetic maps. And so this is the type of thing that it talked about how you could use it. And, it's an excellent Egyptian student, an excellent visiting scientist, Ahmed Salam, myself, and then Adrius Boner is one of the great European breeders and geneticists also too.
So, as you say, reviews are highly cited, but this was 2020, 500 citations, that's 100 a year. That is enormous. How could there be that many breeders interested in citing this?
Well, I would say what probably happened here is, and we should give you some figures. In the U.S., there's less than 120 wheat mills.
Okay, there you go.
Okay.
That's why I can't believe it's such a big number.
Well, so what happens is this. I always tell people you have a better chance of being a Major League Baseball player than being a wheat breeder. And that's true. That is true. Okay. And frankly, we don't get paid as well, but we do have a lot of fun and our careers last a lot longer. Okay. This one got cited because it came out at a time, you think about the science. We were very interested in how to improve our breeding methods. And so this GWAS paper was one of the earlier papers. And there's a lot of papers that people have written since then and they've cited it.
In different crops.
In different crops. Oh, yeah, absolutely. Because this one, would be for any of the small grains, but it would also be used in any of the crops. So you could see it in rapeseed or canola or things like that. And so that's what it did it, because again, it was the theory, it was the how you would use it. And the common thread in breeding is that everything is specific to the crop, but the generalities are for any crop. And so that's why that did so well.
All right, then we're going to go to the last one here, your most highly cited at 825 citations. Drought stress tolerance in wheat and barley, advances in physiology, breeding, and genetics research from 2019.
Okay, now if you look at that one, you can see that it's Ahmed Salam again. He was an outstanding visiting scientist from Egypt, and then an Egyptian student. Wood, I think, is also from Egypt. Adrius Boner is. Ahmed worked with me, and he's also worked a lot with Andreas, and so that's how we got on that. That one is a very good integration, and Ahmed is a very good writer for concepts and making it understandable. He's got beautiful diagrams in it. So his interest is itself in drought stress, and he's looked at a lot of things, and he looks at drought stress differently than we do, because in the Great Plains, our drought stress is almost always at flowering, which is the reproductive stage. And so that really affects yield. If you're in Egypt, where he was doing a lot of this work, drought stress is often at germination. So in the US, we tend not to plant if there's no moisture. But they, as hot and as dry as it can be in Egypt, you can plant with irrigation. And if you lose your irrigation, now what do you got? So it's a completely different type of thing. And what he brought in was, we always did the breeding and genetics, but he also brought in the physiology. So that people, it's not, again, most people, you have a lot of correlative scientists. Like you can say, well, I'm doing high throughput phenotyping and I see this line is this, and it has higher yield. And the question you really want to ask is, why? Because you can have lots of things that look okay, kind of make sense, but you really need a physiological basis. And that's where he came in, his team. So those are, and that drought is such an important aspect.
And
I
think people are getting interested in drought, or way interested, right?
Well, and then the other thing you have to look at is that only in America do we have a crop hierarchy the way we do?
Mhm.
Okay. So for example, most of the world doesn't have the crop insurance.
Mm.
So if corn is risky, you move to a different crop.
Mm.
Okay.
Mm.
Wheat is more drought tolerant, more flexible
Mm.
than corn. Okay. So, and just to give you a perspective, there was one year where we cut, I think it was 55 bushel wheat at North Platte. State average was 38 bushels per acre. So it was a tremendous yield. And the corn yielded nothing due to a drought. Okay. And the farmer made more money on his corn.
I bet.
Yeah, now, why was it? That was due to crop insurance and how the system is done. So, I talked to one of my friends at wheat, and he asked, what was the yield for the state yield for wheat? And for a long time, it was about 38 bushels, just about 2.5 metric tons per hectare. And he looked at me because he was the Senate lead wheat person. He says, "Why are you growing wheat?" And I said, "Well, what would you normally grow?" He said, "We'd be growing barley at that yield level." And I told him, "We can't survive the winter." So we didn't have a choice.
Mm.
But if you were working on just pure free market, our systems would be different. And that's why. So if you look at drought tolerance, people are already, when they're into this area, if they're in Africa, they're looking at the sorghums and the millets. They're probably not looking at corn.
Mm-hmm.
In the temperate zones, it's looking at wheat and barley. And that's probably why it got so highly cited.
Yeah, a lot of citations on that one.
Well, now the funny part is, if you look at those, not one of them is a variety I've ever released. And so Arapaho, which was grown on 1 million acres, doesn't even make it because again,
It's real specific.
well, it's not that, but why would you cite Arapaho unless you used it in a study? There's no reason to cite it. The reviews are the ones that get the citations.
Right.
It saves people time. Everybody can say, "I cited the review. I don't have to repeat it."
Sure. So yeah, what papers do you remember that were good but aren't among these most highly cited? Which ones
Well, the ones that
do
always
you think of the
intrigue me-
most? Yeah.
Well, the ones I think of, the chromosome substitution ones, the anthroculture, the double haploidy ones, the genomic selection, because that really got into the interface of traditional plant breeding, what we could do with new tools, those types of things. And then I've always been impressed, and again, it wouldn't be necessarily, when you work in a team, everybody has a role, okay? And so there are things that are curious that really surprise you. And again, you don't know if you've ever explained their importance, okay? And the one against me is John Watkins, who preceded Stephen McCool, had a number of papers on leaf rust. And he would check every year the virulence of what were the common races in Nebraska. And he never found a race virulent on LR16. That doesn't mean anything to you, but LR16 was a gene for leaf rust resistance. It was initially found in Texas. It was then used by John Schmidt. He put it into varieties, Brule, Redland, became important in Arapaho, Millennium, whole series of wheats. So we had used it from the 1980s through 2000, 20 years, okay? In Texas, that gene broke, which meant that there was a race of rust that attacked, became virulent on it. And so Texas completely got rid of it because it was susceptible. And to this day, to the best of my knowledge, that gene is still resistant, no virulence on it in Nebraska, despite being on third to half of our acreage for years and now probably still present.
Right.
And what it tells you is that there must have been a fitness deleterious effect on the race of rust. So it was the only thing they had, the virulence gene, you found it. And when it was widespread in Texas, it was huge, okay? And they completely lost it. But when they got rid of it, the population lost that virulence because it costed something.
Right,
And that's unusual because if you look at virulence races in leaf rust or many of the diseases, they're neutral. And so you can carry virulence genes for years and years and years. It's almost just by random walk that they get lost, not this one. And so it became an extremely important resistance gene as long as it never did anything in Texas again, which they didn't do, which was very fortunate for us. So I mean, that's the kind of thing where I talk about the curiosity of plant breeders. Why does it happen? How do you explain these things? Right? How do you get to where you are?
So I'm just going to throw out statistics. You probably know these, right? So within the introduction of an antibiotic,
Yeah.
in average, 7 years later, the first antibiotic resistance shows up.
Yeah.
Every single antibiotic that's ever been introduced, about 7 years later, you get some strain that's resistant. Same thing with herbicides.
Yeah.
But you're saying this one
Okay, so
has defied the odds
well, so
in Nebraska.
you brought up a good point. The average life expectancy of a rust resistance gene, there's three major rusts, stripe rust, stem rust, and leaf rust. Okay. Is 5 years.
Okay.
You release it, it gets widely grown.
Yep.
You got, probably trillions of
Electric
spores.
pressure going on.
There's going to be
Opportunity.
yeah, one that doesn't. There are a few that have been remarkably durable. And the one that's along that line is SR2. Second stem rust gene identified. It's identified in the 1950s, and it's never truly broken. Now, it's a unique gene because it's only expressed in the adult plant. It's not expressed in the seedling.
Mm-hmm.
And a lot of other things. And it's not perfect, okay? But it's never broken.
Yeah.
And it's true even in the most, we have a new massive world race of stem rust, which is called UG99, and then all of its morphs, which is absolutely critical. That one still hangs in there. So there is durable resistance in the disease resistance.
Could be the variability of the life path, the expression, and the rust when it, I don't know.
It could be, but the other thing that's interesting is what they think may have happened is that in the pathogen, that it evolved. And if you could go back to an earlier stage where the pathogen has evolved past it,
Right. Right.
maybe you could, sort of recapture the past without harming the future.
Right. Right.
And so a lot of people in plant pathology and genetics are looking at the genetics of the pathogen to see if there's an Achilles heel that evolution has presented itself. We don't know about.
Yep. Absolutely. So as we've talked about, being a professor is a complicated thing. We do a lot of stuff, but also interact with lots of other people. So tell me about any editorships that you've had, patents that you've made. You obviously these varieties are a whole deal. Any companies or any books?
Okay, so let's start with the first one was
The editorships.
editorship. So I've always been lucky. Okay, I always got the easy job. I've been an associate editor for Crop Science, and I've been an associate editor for the Cereal Research Communications. The last one is a small journal, but I love it because it's so egalitarian. It's the one where your English doesn't have to be perfect, your replications don't have to be perfect. And it was free.
Oh,
And it was published in Hungary, and it was specifically to allow people to publish and communicate.
Mhm.
And it's an excellent journal, and it's getting better and better and better. It's getting a nice impact factor. The reason why I say that I've always been lucky is I went from associate editor to editor, and the intermediate step is technical editor.
Mhm.
And that's a really tough job.
Yeah.
That's really tough. The editor job is not bad at all. Associate editor is not bad, but the technical editor, that was huge. So that was the one that I've been involved in. I've done lots of reviews for all kinds of other journals. Got lots of friends that are editors, and they asked me to do a review. I will do that.
Sure.
So that was the journal. So what was the second part?
The next one, any patents?
Okay, now this is always an interesting thing because when you think about plant breeders, and I'm just going to put it in perspective, there has not been a real plant breeder inducted into the National Academy of Sciences since Don Duvik. That was probably in early 2000s. And there's a reason for that. And the reason is that it's kind of like there's a National Academy of Engineers. And there should probably be a National Academy of Agriculture. Because if you look at the science, plant breeders are hard to evaluate. Okay? In that we make our living on our varieties, which there's no citations.
Right.
I mean, the citation or the popularity. I mean, I have friends who've won the World Food Prize, which is the equivalent of the Nobel for food security, you might say. And I doubt they're in any Academy of Science. Maybe in India, if they're from India, but generally not. So the reason why I bring it up is that plant breeders don't do patents. We do plant variety protection. And it's different. And the reason why I smile about that is one of my friends who is in engineering asked me once about, as a member of the National Academy of Inventors, which is the new one they just started.
Yep.
What do you think about it? And I said, well, actually, I don't think about it because I'm not a member. and he was kind of startled. And I said, in this case, my friend, I said, they only recognize patents. They have never pretended to recognize intellectual property.
Right.
It's only patents. And the intellectual property we use is plant variety protection. And the reason why we use it is it has certain benefits for breeding that the patents don't. So, for example, in a patent, you disclose something and how to make it, but you don't have to give the actual product. Plant variety protection, you stipulate that you will be protected for now 20 years, was originally 17.
Okay.
But you will put that material in a collection that will be available for use. And you have your choice, either immediately or at the end of the 20 years, but it has to be deposited. So if someone were going to build upon your invention and its genetics, and the genetics are critical, you have to do that. So that's why plant breeders, I prefer the plant variety protection. And the University of Nebraska was always gracious. As soon as we released it, put it in the collection, anybody could use it. Nobody had to wait the 17 or 20 years. But that's because I think we have a very good university. So I have 0 patents.
Yep.
I have probably 40 plant variety protection certificates, but 0 patents.
That's what I'm talking about. I learned something here.
Yeah.
Companies? Did you start any companies?
I know I never did. And there's a couple of reasons for that. One is that
Sounds like there would be pressure for you to do that.
No. Again, and I'll explain to you a little about me. So I worked in a company. I enjoyed it. I decided it wasn't where I wanted to spend my career. And so the idea of working in a company was never, when I came here, I was very pleased. I was very happy to be here, and I've been happy my whole time. And so I never wanted to start a company. I didn't ever do, because 2 things. One, the capital investment would be huge. Number 2, if you really wanted to do that, you'd probably just go to a company, and I've been to one. And so I'd already been there, done that kind of thing, and it wasn't for me. Now, a lot of really good plant breeders did go to companies. Charlie Brim was one of the greatest soybean breeders in history, and he later went to Funks and Simagagi. And there's a lot of stories like that. But it wasn't for me. So, and again, what we did do, though, was we were very fair with companies. There's a lot of hometown pride, and some states had a reputation that when you crossed their state line, you were persona non grata, unless you were part of their team. And we were never that way. So if you look at one of the proudest things we did, is we actually worked with Bayer Crop Science when they wanted to get into wheat. And we developed a relationship with a company that honored the University of Nebraska's philosophy and allowed them to get into wheat. And so, the story is you can buy germplasm, but you can't buy time.
Mm-hmm.
Right. So when they were out wanting to get into wheat, they went around and they had, I think it was something like 13 first dates. And only one university eventually got into the collaboration. Then eventually they added a second, third, but we were the first.
Mm-hmm.
So we never had to form a company. But if you talk to New Tech Ventures, I think we were probably one of the more successful collaborations with a company that the university's ever had. And in fact, the professorship, my Nebraska Wheat Growers presidential chair, that was a donation from Bayer Crop Science. We can't discuss the full details of the agreement. I mean, even it's funny because Bayer had to get rid of their wheat program when they were merged with Monsanto.
Oh.
And then BASF had bought it, then got it down the U.S. side. But anyway, but leave it as it may, the endowed chair was from that agreement. And so we never started a company, but I think our intellectual property ended up being extremely valuable to the university. And so that's how we did that.
Does UNL have its own seed bank or maintain any sort of?
So, I mean, we keep.
It's all just international or national.
Well, we keep our seed, but you really want, I mean, it's like, in many ways, I want to be careful I put it, it's we do have our own libraries, but a really well curated collection.
Mm-hmm.
You're much better off working with them than trying to duplicate it.
Sure.
So we keep our own seed. We have a lot of seed that we have acquired because we have a lot of friends. That's the way the world works, is friends and collaboration. And it's funny because sometimes things change. So, for example, we wrote to the Australian collection. We got into hybrid wheat, which to me is still one of the great opportunities. And we were, at the time, well, there was everybody in the public sector was in hybrid wheat in the 80s. They all got out of it. And then we got back in when I had the resources and the time and the interest, because I think you can learn so much from it. Okay. And so we got into it, but we wrote to the Australian germplasm collection. And they sent us Peter Wilson's Restorer lines, which it's a gift. I mean, this guy was a tremendous wheat breeder, all in that case with Cargill at the time. And he donated, when they shut the program down, the germplasm to Australia.
Mm.
So we got all the germplasm and no problems. Since then, the Australian collection has put on what they call a standard material transfer agreement, which really restricts how it can be used. So people would come to us to get the seed they should have gotten from Australia because of the way they screwed up the intellectual property around it, which bothered me because it was still a gift. I mean, the Australians, it was a gift from Cargill.
Mm-hmm.
And now you're charging money to distribute it, which is why Cargill gave you it the gift in the first place.
Nope. I've read all about seed companies protecting all of the seeds.
Well, see, they they prefer patents.
Yeah.
And they do the patents because, again, they're not as interested in, doing quite what we would normally do. That sort of thing. It's a different perspective.
It is. All right. Which organizations funded your research and how did that change over time?
Well, the main one was the U.S. Department of Agriculture.
Okay.
So, a couple of things.
So you write a proposal and then it gets funded, or there's some other mechanism?
There's people at a land-grant university often don't understand the origin of the land-grant university is one thing. They secondly don't understand necessarily how you're being funded.
Mm-hmm.
So there are funds which come to the ag school, Tiffany and the Institute for Ag and Natural Resources, that are called hatch monies.
Mm-hmm.
There are monies that come from Smith Weaver. There's those types of things. So we had funds from those. And so, people I think are very jealous of that, but it's part of the federal expenditures. And used to pay for salaries, technicians, things like that. But then you could never live with that. Okay. So the most consistent grant money we had is because we work with the Nebraska Wheat Board. They have a checkoff. And so they were the ones that funded the varietal improvement. Okay. Then when they funded the varietal improvement, if we wanted to do curiosity research, we didn't think it was fair to ask them to do that because they needed varieties. That's how they got their checkoff student. So then we would go to other organizations. Usually it was the USDA. Usually it would be the AFRI and NEFA and those types of things. We did a lot of multi-state work with good collaborators at UC Davis. George Dukovsky was the lead and put multi-state programs together. We got funds from there. We did a lot of work initially on international grants. So the BARD groups, which was, I think, Binational Ag Research Development Grant between Israel and the US. And then what most people...
So those would be funds from those governments or from the United Nations?
No, it'd be from those governments.
From those governments, okay.
From those governments, which that's a good point because I'll bring up another thing kind of that. So we used to do that quite a bit. Then a lot of my friends in Israel retired and I didn't really pursue it as much. We were always very good, or very open at attracting scholars from other countries. So if you look at my international students, virtually every one of them came on their own money. So it would either be a scholarship, now maybe it was a US Egypt scholarship or a Thai scholarship, or in Turkey, it was actually a German scholarship for Turkish students to study abroad. So we had, I don't know, 25 foreign students all on their own money.
Wow.
Well, you know,
That's pretty good.
well, it was, they knew we were open.
Sure.
And that's something which I think people forget. And we always treated them well, I hope. I mean, you know, maybe a couple that didn't think so, but by and large, most of them came, got a fantastic education from the University of Nebraska, went home and they're succeeding very well. So, you know, when you look at that, I mean, you know, you figure that it wasn't a GRA cost for $35,000 a year. And we had people, one from Philippines, and she spent 4.5 years here. You know, so that's what, 200,
Yeah.
150, by the time you had the overhead, $200,000 grant. Yeah, we had many students from Thailand, many students from Turkey, had a lot of CONACYT fellows, which would be the Spanish equivalent from Costa Rica, Mexico, those types of things. The MEAG project had a lot of Moroccan students. So we were very fortunate that way. Indirectly, we didn't apply. And then we had a lot of Fulbright visiting people, which were wonderful people to have too.
I don't think of Thailand as having a similar environment as Nebraska.
Oh, no, they didn't go back to breed wheat. No. They didn't go back to breed
You're all
rice.
learning how to breed. Yeah.
They learned how to breed and they went back, you know, and I have to laugh. The one that went back to the Philippines, I think, is working in coffee, as I recall.
Oh, okay.
Yeah. So yeah, no, they all went back, but they got an American education, and that's what they wanted. Yeah.
What techniques and instruments did you use to do all the breeding? Techniques and instruments?
Well, okay, so in a breeding team, it's always what's available, what answers your question. And so a lot of people, you know, some people I think are what I would call technology astute, but they forget that if you can answer the question, that's the key point. So we always tried to keep our equipment current. And the reason for that is that I think a lot of people look at agriculture as sort of backwater. You know, it doesn't have scanning electron microscopes. It doesn't have X-ray. It doesn't have Y. It doesn't have robots. And well, in fact, it does, because all of our genomics was done with robots, okay, and things like this. And all of our phenotyping was done with drones and that sort of thing. But I never wanted my student to come to the field and use a combine. We actually had the 4th earliest combine imported into the US for small plot research.
Wow.
It was easy to fix, don't get me wrong, and it did its job. But that's not the impression you want. Your impression should be that you're using very good equipment. And like, so we always try to do that. We always tried to have a truck be no more than 8 years old. Now, we didn't use them a lot. So what we do is we always have two big trucks. And that's because they can pull these huge combines, which are tiny compared to a farm combine. But we always wanted to have appearance that you were working in a project that was worth investing in. And so that's what we did. So, we use the small plot combines, and I'm just impressed because my successor, Catherine Frells, is doing really, really well. She's taken the combines we had now. Now, what is a modern combine? Well, a modern combine, cut a plot, it'll weigh it right there.
Yeah.
It'll give you the test weight. It'll give you the moisture.
Yeah.
So that's all things you'd use for correction. It'll give it on a printout, which you can download to a computer. Okay. And she's now added an NIR so she can do protein at the same time. Okay. You can subsample, so you don't have to keep the whole bag if you only want to keep 1,000 grams instead of 2,000 grams. So that means that you have much less storage. All that's being done. It's fantastic.
Yeah.
And, if you go to a seed company, which again, you don't want to train your students for a technology that no one uses it.
Exactly.
They'll have it set up. They'll put an algorithm in and they say, okay, if it's 1,500 grams or less, just put it in the bin. We don't even want to keep the sample. If it's 1,500 or more, we want 100 grams or whatever they need. And if it's this, they'll do something else. And they'll have that right and they'll bag it separately the way they'll do that. So you want to have that. And then you also want your students, every student that I've educated, the companies have told me they appreciate that they're hands-on breeders. It's easy to get people, it's kind of like you had a summer job, you want to be a bank teller, air conditioned, nice thing. Okay. Well, we're out in the middle of the heat and the dust and all that kind of stuff. So they will appreciate that because that's what breeding is.
In Nebraska, no less.
Everywhere.
I know, but it's hot.
But the point is you want to be,
Yeah.
and in fact, the best people in that field are the ones that can do that. And unfortunately, it's very common for people to become allergic to pollen.
Oh.
And so you have to be up. I had a friend of mine that got a serious disease, and the medication was one that was a sun poison. So if you went out to the field and you got the sun, it made, I think, super radicals. And that ended his field career. He was a field plant pathologist. He could never go to the field again. It didn't matter how many clothes he put on, how much shade, he couldn't go out. So you're lucky if you don't get things.
Wow.
Yeah.
So in some ways, science, and I'm sure agriculture, is cooperative and competitive.
Yeah.
And did anybody ever work in the same fields that you're working in? And I don't mean by fields, but and did they publish at the same time or counter you?
Yeah, but we never got scooped.
Okay.
And it's a field of gentle people, men and women. And the thing that I think that always I appreciated was that to publish a field study, you have to have at least 2 years. We often had more, at least 2 environments. So if you came up with a really good idea and you're testing it, you could tell the idea when you finished and say, this is what we wanted to do. And if someone wanted to replicate it, it was 2 years and 2 environments. If they had the genetic materials, which they probably didn't have either. Okay. So there were people that I was aware of that got scooped on how to use molecular markers and things, but that never happened to me. And again, that was probably because the people that were getting scooped were the ones that were creating new things, not necessarily applying them. And so it's easier to scoop someone on an untested idea or like a marker or a biochemical type of thing than it is to actually scoop someone on, hey, this marker really does do this. So that never happened. Again, it's a field of collaborations. The thing that has hurt plant breeding more than anything is the restriction of access to germplasm. And we were always very open. And there were companies, I have very good friends that worked at companies. The company had a strict policy. They would not share any of their germplasm. And I'd still write to my friend and say, hey, we're going to release this line. When it's released, it goes to everybody. Would you like some seed of it? And you can use it, it's unrestricted. There's no pass-through rights, anything like that. Sometimes the company would still tell them no, because they thought there could be a risk. But quite often they would say, well, thanks. But, that's why we're at a public university. Right. You can do those things.
So the companies, they're trying to make money on a particular product, and you've got lawyers who are trying to protect their rights. And one way is don't let anything go, right? So the lawyers are always going to tell you, don't give anything away.
Yeah.
But you are not restricted by consulting with lawyers. You're more in the academic, let's do this.
We have our tech transfer consulting. Yeah. But I think the point that comes out of that is I work with a foundation now. And one of the guys in the foundation is a very clever man who used to work with Bell Laboratories and AT&T and that group. And what you'll discover is companies have very good ways of working together. And so it's a little bit, he'd come up and he'd say, well, and he invented the two-factor verification. He and others, he's one of the inventors. So, he's good at it. But when you get into a software issue where there's all kinds of things, they just write an agreement that says you can use this and we can use this, and it gets taken care of. Because the risk of the lawsuit is probably more. And if they got into that deal, they would lose access to what everybody needs. And that's kind of the way the germplasm is. There are some people that won't share. No, that doesn't work. And because of that, we got germplasm from almost anybody. Unless they had a strict policy, well, Steve, he shares with us, we'll share with him. Other people didn't share with them, well, then it's reciprocal. That's right.
All right, well, let's move on to other career activities, like societies. Which conferences did you attend regularly, and was there a meeting that was especially memorable?
Well, the one I always attended was the Crop Science Society of America. That's the one I always call my home. And I went there as a graduate student, and I became eventually the president of the society. Then I was a division chair, all kinds of stuff with that. Editor-in-chief for their publications, these kind of things. So that would always be the home. And I remember the first one I went to because I won a prize as best graduate student speaker. Other than that, it was always enjoyable just to meet your friends and see what they were up to and that kind of thing, the sharing and life. So those were always memorable. I enjoyed the plant and animal genome conferences because that was something we went because we had grants and the grants would meet in conjunction with them. But it was always interesting to see what other people were doing, to listen to, and I can't think of his first name, but I think it's Leroy Hood.
Yeah.
And he talked about his team of 100 workers and postdocs and whatnot. 1/3 were engineers, 1/3 were biologists, and 1/3 were chemists. And describing how a 3D printer worked remarkably well for synthesizing DNA and for putting it in spots, It was just amazing. So that kind of thing I have enjoyed. Yeah. Every meeting I ever went to the American Association for the Advancement of Science, I enjoyed for the same reason. Not because my field was particularly highlighted there, but boy, you could see the diversity of science and that. And again, that's just the curiosity that a scientist should have. So I was lucky at that. I worked with the the group that publishes bioscience. It's the American Institute of Biological Sciences. I was an ad hoc, I had a large member there. Was a division member in Section O of AAAS. So there's other ones I've had leadership roles in, but the one that's always home is the Crop Science Society and the American Society of Agronomy.
How about sabbaticals? Did you go on any, and then did it change your direction?
Okay, so to me, this is interesting because I never did a formal sabbatical. Okay, and the reason is plant breeding's a hands-on thing. Well, no, you think about it. We made our crosses in March. We take our field notes in May. We do our harvest in June, July. We have to set our nurseries up in August. We have to plant in September, October. And I actually put another crossing block in because speed was important in December. And so January and February were fairly open. Okay. Well, it's hard to get a 6-month sabbatical in 2 months, right? So I did do there's a term for it. It's a temporary leave of absence or something like that. So I did do 4 months, and I went to Australia and worked in a wonderful molecular biology laboratory. And it was outstanding. And I would recommend, the other thing I should mention to you is I've been very fortunate. Because I've had so many international scientists, I get invited to a lot of international meetings. Okay. Because I've had a lot of Fulbright temporary scholars, I get invited back to their country. I used to work with the International Atomic Energy Agency on some of their biotech issues. I would visit those countries. So I've been to a lot of different places. To me, that was always the valuable part.
That sounds good.
And the leave of absence, that was fantastic. I could have learned probably how to do it, but to change, see a different culture, see how they work together, work with one of the great molecular biologists of all time. His perspective, because he started in Dressala and then moved to wheat, and he kept projects in both for a while and then ended up only in wheat. The team around him, they were excellent people. State of the art, absolutely state of the art. And like any good program, they rid your mind of a lot of sort of misconceptions. Things like, do you have to store your DNA this way? And he said, well, actually, you can put it in distilled water to work just fine. Things that if you read, you would never find out.
Yeah.
See all the shortcuts.
Yeah.
Okay. So I highly recommend it. I think people don't do it nearly enough. I probably didn't do it nearly enough, but it's funny. I went on one, and the next person I know that went on one was James Schnabel. And he called me up, and again, he couldn't take the 6 months. I explained to him how I did it, and that kind of thing. And how do you handle your responsibilities and that sort of thing? And we got that all done. And I think the university, to my way of thinking, doesn't support sabbaticals as well as it should. Some departments will tell you that you have to find your own replacement. And in an employment contract, that's not right.
No.
I mean, if you're supposed to have a sabbatical, that's an institutional responsibility.
Yeah, that's unfair.
Yeah.
How about service? What service contributions do you remember the most to the department, the university, to the community, to the world?
Well, okay, so when it came to service, I was the interim department head for 2.5 years.
Oh, there you go.
So that probably counts. I was what I always thought was interim associate dean. I think Tiffany said probably an associate dean of a limited time in Casper. So those would be the big ones, and I enjoyed both. Not sure I was really good at either, but they all seem to have liked it. I really enjoyed working with the International Atomic Energy Agency. And the reason for that is they often sent you to places where agriculture was important. Okay, that's the first thing. Secondly, if they got the agricultural funds, it meant that they could inspect anything they were doing with atomic energy. So if you think about it, like Iran, right? They want the inspectors back in.
Mhm.
Well, okay, if they do that, then Iran's going to get agricultural. I mean, there's 5 legs of the Atomic Energy Agency: medicine, agriculture, nuclear power, and 2 others. I can't remember what they are. Well, there's parts of the world that you would get to see that, you wouldn't have the opportunity to.
Mhm.
And I always enjoyed that. And they had what were really trivial sums of money. They were $5,000 a year. And that was probably now much larger, I hope. You go down and you review a project. But I went to Peru, and the guy, Peru is dirt poor, lots of government issues, and that was a lifeline. Then to Ethiopia, the same way, that was a lifeline.
Mhm. Mhm.
And you got to see really good people
Mhm.
working in very harsh conditions, financially and whatnot, but doing their level best. And so it was perfect.
Mhm.
Yeah.
Sounds good. Tell us about the courses you taught more often than others, and then what are your thoughts about teaching those courses?
Well, okay, so I only taught 1 set of courses.
Okay.
There was always the introduction to plant breeding.
Okay.
Well, I should be careful. I did add a few others on. There was a time when I thought that teaching in plant breeding and genetics, the University of Nebraska at one time was stellar in the area of genetics. And at one time, I think 20% of all the instructors in genetics that come through the University of Nebraska. I mean, it was an unbelievable number.
Wow, I didn't know that.
Yeah, no, I mean, we would be, well, it's
Is
not
that from
like
Kayam?
Kime was an incredible instructor in genetics. And, Kime's son, Wayne Kime, so we're thinking of Franklin Kime. Wayne Kime learned genetics at Cornell and a lot of things, but, they have been the undergrads that came up and then went elsewhere to get their
Yeah.
graduate degrees, but we had a huge impact. And we've been lucky. I mean, we had Dave McGill and Don Lee, and I have a new professor. I think they'll all be stunning, great teachers of genetics, in fact. But at the graduate level, we lost a lot of our skills. You know what I mean? And it's part of it's evolved. I mean, and I shouldn't say we lost our skills, we've changed our skills. But the actual aspect of plant breeding lost some of the courses. So I was concerned. So I always taught the introductory course. And we did something which was a little different. We broke it into 3 1-credit modules. And we did that specifically because I had a student who got called up in Gulf War I, not as a graduate student, as an undergrad. And he got called up in November. He lost that semester because he couldn't finish it. He was in Iraq for a year, and he came back in February, and he lost that semester. So functionally, he lost 2 years. And the university at the time didn't have a way of accommodating.
Yeah, right.
So I said I never wanted that to happen again. So I had a self-pollinated crops part, a germplasm and genes where you get your germplasm, and a cross-pollinated crop part. And they were each, I guess, 5-week modules. I think that's where a 15-week semester. Each was a 5-week module. And we did that specifically so that no student could ever get caught that way.
Mhm.
We always taught at resident distance as soon as we had the capabilities to do that. So that if you're out in the field, you could come back and hear the exact lecture that you would have had if you were in
I
class.
see.
There was just a lot of things like that.
Mhm.
We did all of our testing online
Mhm.
because I didn't want them to be time tests. Many of our students speak English as a second language. I didn't want to grade them on English. So we tried to make it as accommodating as possible.
Mhm.
And I think it worked.
Mhm.
I really do. And so I taught the introductory self-pollinated
And
crops.
what was the enrollment in that class?
Well, it started out, depending on the year, we got as high as 40 students, if you can believe it, which blew me away. I mean, usually when I was teaching it as a resident, and again, as departments ebb and flow, if you have a lot of genetics people,
Mhm.
then they all put them into that class because they all need to be able to communicate with plant breeders. So normally we would be averaging probably 10 to 12.
Okay.
But, some years you'd be as down as low as 7. And so, but you wanted, and again, if you did it only resident, there'd be 4. So that's the other reason for doing it. If you really love teaching and you want to make it, and the biggest honor of all of that was when a friend of mine sent a student from the University of Illinois, which has a good breeding genetics program, but it didn't fit the student's schedule, so they had to take a distance and came to Nebraska. It just shows you the capability. So we taught, I think, from 2008 on, something like that, distance.
Oh, yeah.
Yeah, I mean, we were one of the early adopters. We were on VCR tapes.
Wow. Yeah. Nope. All right, let's move on to your youth. Tell us what it was like to be a young Stephen growing up in Des Plaines, Illinois.
Well, we were, it was I guess I laugh because, in the day, education was considered very important. My father apprenticed as an accountant. My mother graduated from high school too soon to go to college, so she went to secretarial school. So neither one had a formal degree, but they both valued education immensely. I always loved science, and so I was the kid that woke up at 5:30 in the morning to watch the science studies, on TV in the morning and the like. And there were hilarious things you learned.
Mr. Wizard?
No. This was educational.
Okay, wow. Yeah.
So it'd be like chemistry and things like this.
Wow.
But the funny part was that,
In Chicago area, I suppose.
Yeah, it had to be. When you're not watching Bozo the Clown, you'd be watching this.
2 choices.
Yeah, well, no, I mean, Bozo was always that was the big thing. But the funny part was, you got this. I mean, I don't know if people can think about this, but like, I remember being in high school in physics class, and the teacher saying, this is not a very professional tape. And so the guy gets a coughing fit, and they just move the camera away while you listen to him cough. I mean, they didn't edit it out. They didn't do any of that stuff, okay? Well, in the morning, they had this guy that was on chemistry, and it was like practical chemistry, and it was amazing. It was like, what is the color of ketchup? It's brown. And it's only red because they had red dye to it, because that's what you expect it to look like. And what's the color of ammonia? It's clear liquid. But they had a whiteness so you don't drink it.
No, I love these kind of things. Yeah.
and this guy who's giving this lecture or talk or whatnot, engaging us all talked about the time he made a stink bomb, dropped it out the chemistry class window, and then he watched, much to his chagrin, one building after another. It must have been really good.
Yeah.
Evacuate. I mean, it's these kinds of stories. It was funny. And I always loved being outside. And where I grew up, there was a farm right behind us, which we used as a playground, much to the dismay of the farmer. And there was an area further down that had was small, a lot of just sort of rough grass kind of thing, torn up, sort of a construction site. So I always loved the outdoors, that kind of thing. I was familiar.
Probably a mall there today.
Not there, but there's a lot of other stuff where the farm is.
Yeah.
And the guy was an amazing farmer because, he grew tomatoes for Campbell's soup.
Oh, okay.
He grew corn. And the whole suburb was basically bought out of his farm. And he took, I think, 5 cases to the Supreme Court on tax law and won all of them.
Wow.
Yeah. The way he made money was selling that farm. People would try to rezone it. The city wouldn't rezone it. They'd go bankrupt. And he had written in the contract the land is reflected back to him. And he'd sold it numerous times to people who had wishful thinking. Finally, they made an absolutely gorgeous park out of it. The city just bought the land, made a park out of it. It was perfect.
All right. So then, how did you decide to go to Harvard and then major in biological chemistry?
Okay, so I applied to 4 schools. One was Harvard, one was Brown, one was Carleton. My brother went to Carleton College in Northfield, Minnesota. The other one was the University of Lincoln, University of Illinois, because I was in Illinois.
Yeah, yeah.
And that was sort of the one I assumed. I was the valedictorian of the class, so I assumed
Wow.
that I would get into that one pretty much for sure. And didn't know about Harvard. Well, Harvard had a history of taking the valedictorian from the class. We graduated 800, so it was a big class. And so
For that time, what kind of year we talking about?
I graduated in 69.
60.
We were the baby boomers, there's no question.
Yeah.
And I got accepted into Brown, I got accepted into Carleton, and I got accepted into Illinois. And I went to Harvard because a couple of things. I drew a 500-mile radius around Des Plaines. I wanted to be on the outside. So that was Brown or Harvard. I liked Harvard, I liked Brown, but I liked Harvard a little better. I went into biochemistry because my original goal was to be a human nutritionist. So I always wanted to work in feeding people. And, there's some great quotes. I even brought some of those with me, which, I don't know did you ever hear the book, The Population Battle?
Yeah.
Okay. So I'm 18 years old.
Yeah.
The battle to feed all humanity is over. In the 1970s, the world will undergo famines. Hundreds of million people are going to starve to death in spite of any crash programs embarked upon them.
Did you read that in 1968?
68. New York Times bestseller, man, National Academy of Science member, all that. Okay. And there was no way I wanted to live in that world. I wanted to do something to fix that problem.
Yeah.
And of course, what they didn't know is that when Warlock won the Nobel Prize in 70, there never was a famine, not at the way they're describing it. And the great families that were described there were earlier.
Right.
There's the partition of India and Pakistan, and 40 million people died.
Yeah, Borlaug is the counter to
Yeah. And so that's what got me into it. Well, I got there and I was in biochemistry because it was the nearest thing to prepare you for human nutrition. And I don't want to have this sound badly, but biochemistry is also the pre-med major. And so I was fortunate. Organic chemistry was never an issue for me. Physics, the biochemistry, that was, all the, not easy, but doable. And all my friends that were pre-med majors were just sweating everything, couldn't understand that. And they were frankly not people I wanted to associate with. Okay. Well, at the end, I moved away from human nutrition into plant physiology. Mm-hmm. And there were two great professors there. One was Winslow Briggs, who discovered phytochrome. The other one was Lawrence Bogerman, who did a lot of work on porphyrin rings, which is what you have in photosynthesis as well as in hemoglobin. And so I got interested in plants, and they wrote letters of recommendation for me. Mm-hmm. And I applied to 4 schools. Mm-hmm. So that's how I got to Harvard. Mm-hmm. I had a good high school, so I came in as a sophomore. I spent 3 years there. I didn't want to spend the 4th because I was ready to get on with things. I actually applied to the Peace Corps at that time too, but they were so slow that I already been accepted into graduate school, and the graduate school was more interesting. So that's where I went to graduate school. Anyway, I applied to 2 schools of breeding and 2 schools of crop physiology because my background would let me go either way. And it ended up that Purdue University offered me the highest paid GRA, not that was a big deal, but it was sweet, single guy. And it was on high lysine corn, which was right at the, it's a quality protein maize is what you call it now. That's what Brian Larkins did his work on. Mm-hmm. And that's where I went. And it was always to feed people, always to feed people.
So that's how you chose Purdue. And then what actually did you do for your graduate work?
I worked on what would be called quality protein maize.
But you learned breeding, or you?
Oh, I was breeding and genetics.
Breeding
and I always learned the breeding part. That's what the major would be.
And that's where you learned breeding.
Yeah.
Okay.
Yeah. Harvard had no school.
Yeah, exactly.
It's funny. I did take their economic botany course at Harvard, And it was taught at one time by Magna, which was one of the great corn evolutionists. But it was taught by a pharmacognosist, a wonderful guy who searched for drugs, basically. And it was Amazon rainforest, Mexico, learned about peyote, learned about all the medicinal plants of the Amazon, that kind of thing. But he had a really good economic botany course. And actually, one of the TAs that was there later went on and became the small grains breeder at the University of Illinois. Well, I take it back. He was University of Maryland, pardon me. And I had a joint appointment there when I was at Beltsville. But I learned breeding there. But I also learned a lot about chemistry. I mean, the whole thing about quality protein maize was the nutrition aspect of what you eat and how you measure it and things like this. And it was funny because they talked about one of the measurements was how much weight you would gain when you were eating something. And they fed a very low protein diet, and these animals just got to be little tubs of lard. They had no protein. And so they were eating and eating and eating the calories, trying to get the protein, and they just became super fat. So you learn what worked and didn't work, trying to measure nutrition. But that's why I went to Purdue.
Yeah. So how did you choose to go to USDA in Beltsville, Maryland?
Well, so that's an interesting story because, at least to me, anyway, because one thing that happens is at the time, plant breeders didn't have postdocs. And I'm not sure that that's something we really benefit from, to be honest with you. So it was always the idea to be on the job training. So when I finished up, I looked at a job with Northern King, and it would have been a good job, but it wasn't what I was interested in. I looked at a job at Guelph. They didn't offer it to me. That was kind of a surprise because when I interviewed, they said, "Boy, you've done a great job. This job's yours unless some of the other candidates come in." One of the other candidates came in.
Oh,
I wish they'd never said it. I interviewed for a job at Beltsville, Maryland with the USDA. And I interviewed for a job at North Dakota State to be oat breeding. One was oats, which is a relatively minor crop. The other one was wheat and barley, which are truly global crops. So the opportunity was good. The USDA, until recently, was the best place to start. To think about it, they have 2 publications in your first 3 years. How many academics, you would have been,
that's unusual.
Yeah, you'd be in trouble. And they were very gracious about that. They treated you well. You didn't have to get grants. That basically the idea of USDA was if they didn't have funding, they didn't hire you. So it's a completely different culture. It was a great culture to work with. Beltsville was a magnificent site, which again, they're selling. I don't know if you know them, but they're selling that off.
No, I didn't know.
Yeah, so it's this decentralized thing. I don't think they'll ever have the way they once did. But again,
I think the whole point was to decimate the whole USDA.
Well, I don't know if they're going to do quite
But,
that. I mean, they've got these great ideas, but the problem is the scholarship is so poor on the planning. So if you look at it, in 1972, I was there in 1976, okay? The organization in 1972 is basically what they're going to try to go back to in 2026, 2027. And they're going to do it in a decentralized fashion, which a national government never did. So it's like you learned nothing in the last 50 years, or you forgot everything. So that was the kind of the way that did that.
Right, but their whole model has been to rate reports, right? So that everybody shares, and so then, but you've got this whole information pipeline.
Well, they actually had fantastic scientists. That was the thing, G.F. Sprague, who was a National Academy of Science member for corn genetics, and a brilliant scientist in quantitative genetics, was at Beltsville and Illinois. the person who hired me, John Mosman, was one of the first people to actually go from race virulence to, post, avirulence to gene for gene. As soon as Flora's work came out, he went right in there. He did all the genetics of how we mildew.
Mhm.
He had elegant genetics, elegant disease resistance, which actually, I got to tell you a story because this is useful. We talk about publications.
Yep.
Again, plant breeders, we release varieties, we teach or should, and we write papers. And so there was a fellow by the name of Isaac Wall, who was one of the greatest mycologists, fungal geneticists, fungal people, researchers at Hebrew University in Israel. And the origin of wheat is the Middle East.
Okay.
So he and John Mosman worked very closely. And Isaac was this guy who, when he was like 9 years old, he with his 13-year-old sister walked from Poland to Israel on their own. I don't know where the parents were because of the migrations that were coming. And they avoided the Holocaust because of it.
Yeah.
So anyway, he's got 2 students. One of them works on small grains, and it's a catastrophe. 5 years to get tenure, 3 years of drought. They work with diseases. You don't have diseases and drought. What are you going to do? It's got very few publications. The other guy works on mushroom genetics. And so every 3 months, he's got a new experiment. It's just flying. And so Isaac Wall is in there, and he's at the tenure committee. And he comes back, and they're giving my college with the guy on mushrooms, no problem.
Yeah.
The guy that's doing the really heavy lifting on post-plant, post-parasite interactions is getting creamed. Finally, Isaac Wall looks at me and says, at this university, now Hebrew University is, at the time, the university for the Jewish theology. Okay, so at this university, if Isaiah himself, the prophet Isaiah, came up for tenure, you wouldn't give it to him because he only wrote one book. and he got his guy through. But I always laugh because when you think about, how many publications,
Yeah.
it's always one more than whatever you had.
Yeah,
I had years when there'd be 12 or something like that, and it'd be like, well, it's one more than what I had, I guess, So anyway, I always laugh because publications don't tell every story.
Yeah. And Monsanto.
Pardon me?
How did you choose to go to Monsanto?
Oh, that was interesting. I mean, every job I've ever had, I assumed I would be there for life. Okay. And I loved the USDA. But after I was there 7 years, they did 2 things, which were, I found, well, there was a couple of things. Number one is, if you work for the federal government, the president can make a big difference on civil servants, okay? They had a fellow there by the name of Devine was his last name. Probably the most misnamed person to run personnel management, okay? And I remember I lived close enough to Beltsville that I could walk to work. And so we had a snow day where it was like 24 inches of snow dropped. The roads weren't plowed, nothing. But I figured I'm only a mile away, I could walk. So I walked 24 inches to all this snow to get there. And I walk into my building, and there's this lovely secretary, Nadine Hires. And I say, Nadine, how are you doing? And she says, I'm upset. And I've never seen her upset in my life. And I said, why? I came all the way from Lowell, which was like 10 miles from this crap, because I was told to come here. And the head of Beltsville, quite rightly, said, come home. Don't come. If you can't, don't come. If you're here, go home, because the storm is going to get worse. And Devine didn't give a damn about a civil servant. He just absolutely didn't. Okay, well, that was one thing. But they did something else. The person I did all my agricultural research with was a lab chief. And for some reason, I have no idea, it was never explained to me or to anyone else. They said, you no longer be a lab chief. They just put him back as a bench scientist. So here was a guy that was doing what you would call the administrative route. He was going up there, and he was completely just jerked his chair. They had another guy who was the national program leader for wheat, an absolute gentleman, one of the best people I've ever seen in my life. And they decided to reorganize, and they went from 55 national program leaders to 33. Okay? And the way they did it is they basically said, these 22, you're no longer a national program leader, you can leave the room. You 33, you are still program leaders, and we're going to tell you how we're going to do all this. Okay? And I looked at it and I said, I don't trust them to go up administratively, and I don't trust them to go up in the philosophical side either, the strategy side.
Right.
So I said, I'm not going to do this. So I made a decision to leave at that time. And I looked at Monsanto, I looked at another company. I don't think I looked that often at universities. Mainly it was companies. There was a big tech boom back then. And Monsanto was hiring good people, good company. So that's why I went there. And to this day, I will say, when it came to the quality of the individuals, they may be the organization that was A, most representative, and B, had the highest quality, because money wasn't a object at that time.
This was the 80s.
It would have been 1986.
Okay.
1983, 1983. And they had Roundup, they were making money hand fist on Roundup. They had Lasso, they were making money on that. Triolate, which is a small herbicide, but it was making them tons of money. Everything was going. They were getting to buy seed companies. That's why they were interested in someone who could work with a seed company. And I was their liaison.
Yep.
But in the 3 years I was there, less, they had a lot of changes. And when I gave a seminar, and they were at 360 people in the ag division, and they called me back for an interview, because I think they're using the seminar as sort of see if the guy would be any interesting to us. They were at 400, and in that less than 3 years, they went from 400 to 600. They bought Searle, had to cut a gazillion people. And then by the time I left, they were under 400, which is what their target number was, so they had to rehire. So it was this accord. Now they're much better, don't get me wrong, and the Bears probably had even better too, then, because they're very stable. But at the time, that's when I left Monsanto.
The 80s were a time of mergers and trashing other companies by buying them up.
Well, there were things like that, but the Monsanto had a pretty cutthroat mentality, which they then changed. I think they saw the error of their ways, and friends of mine that stayed did very well. But I felt that I couldn't hire a person to come into that situation. You can't hire someone, then you're probably not the best employee either. So that's why I left. That's why I came to the.
So then you came to UNL as an associate professor. Was that with or without tenure?
With tenure.
With tenure. And so how do they justify that?
Well, so it's the story I tell my students. If you want to try industry, it's good to build your reputation before you go to industry. If you go to industry directly, make sure you keep your reputation visible. So when I left the USDA after 8 years, I had 33 publications.
There you go. Yeah,
okay.
That'll do it.
And they said, well, Steve, do you ever want to teach? And I said, well, the USDA actually has its graduate school where they teach courses. And I taught plant breeding for poets.
Oh.
And the reason is there were a lot of people that were curious about what plant breeders did. There were very few of us in the Beltsville area. Most of the breeders were assigned to universities or collaborating with universities on site at the university. But they had all these biotech people, and they really wanted to know how a breeder would think. So I tried to explain them the common breeding methods, these kind of things, enough that they could converse, enough that they wouldn't say silly things. And that was extremely enjoyable. And then They always had their end of the semester banquet in the state ballroom at the Department of State.
Oh, wow.
I mean, so you're in there with, the original portraits of Washington.
Wow.
On a carpet that's got the seal of the United States. It's about an inch thick.
Wow.
I mean, it's like unbelievable.
Wow.
And the food matched.
Mhm.
So, I mean, so it was a good time. I did that for 2 years. So I knew I liked teaching. I knew I liked research. And then John Schmidt, Virgil Johnson, Rosalind Morris, and Paul Martin were the greatest wheat breeding team of my generation. And so when an opening came up, which was actually extremely fortunate, I applied late. The closing date was before Monsanto had just. What happened is I was in Rome at a meeting for the International Atomic Energy Agency. Monsanto let me go there. And working on IAEA stuff, but it was mainly a collaboration. It was a wonderful meeting in Rome. Oh my God, what a beautiful time of year, Christmas! And we were very lucky. We had my unit's Christmas party before I left. I went there, and on Thursday, I was to fly back Friday. I got the call that they'd fired well over half of the whole unit I was in. The division, over half was fired. And the group that I led, over half was fired. And then they added another half, the other group, back into mine. So we ended up about even, but it was like, holy crap. And it was over that time, I made a comment to my wife that this happens again, I'm quitting. And I said, if this happens again, I'll be fired. So I said, screw it, we're going to go find something. And this was an outstanding university, an outstanding program. And they did a beautiful hire. They did everything right. The position was advertised as assistant or associate professor with or without tenure, with my publications. And at the time, I'd had a lot of leadership roles in various different groups. The resume looked good. They felt like they'd be with tenure.
Yep, and you had teaching experience, so
Yeah,
you checked all the boxes,
Well, and it was funny because it so happened that after that, another university called me and said they would really like to have me come. And I said, I don't want to waste your time. Yours is advertised as an assistant professor, and I've already applied for one that's an associate professor. They're interested. They'd already talked to me. And they said, well, we still want you to come. So I did. And it was a great thing. And it was a much larger teaching role, which, it would be 100 students, which I'm not sure I would have been prepared for. But they did a couple of things. One is that while I was there, when I was interviewing, they liked me enough that they switched it from assistant to associate. And it spooked me because I didn't think they had, I didn't think they had internal controls.
Yeah.
And so that spooked me. And then the team was much smaller. The impact was much smaller. The importance was much less. And so that's really what did it. And then the other thing which was interesting, and just to put it in perspective, money has never been the driver. It's always been your passion. University of Nebraska offered me exactly what my bottom line would be to leave. The other university actually offered me, about 20% more. And then it added summer salary, which meant that they're actually going to pay me more than what I was making at Monsanto.
Wow.
And
They wanted you.
well, I guess so. I don't know. Anyway, but it still wasn't right. And so I came here, and the thing that it did do was I wrote back to the UN and I said, you've treated me really well. You've done everything properly, but I just got to let you know, I got this offer. Is there anything you could do a little better? And they wrote back and they gave me 3,000 extra. And it was perfect. And then that was the year that Kayor was the governor. And we had 3 years
Good times.
3 years of massive increases.
Really good times.
So that really came out. So it's the thing. But it was the reputation of the program and the reputation of the school that really made the difference. And then the opportunity was, I'm glad that I was late. I'm surprised Yeah, they must have had in their closing date or until a suitable candidate's been found. And I applied. I guess they thought that was a good deal.
All right, well, let's move on to your family. We just learned you had a wife. Do you have any kids? And then how do they fit into this narrative of moving all around?
Okay, so this is one of those things. I have been married twice.
Okay.
So I got married and have two kids from the first marriage. And they moved, we got married after I'd gone to Beltsville. And then we moved to Monsanto. And then moved to Lincoln. And we grew separate.
Mhm.
And so my wife and I split. And my two kids were with her. And then 3.5 years later, I married my current wife.
Mhm.
And she has two kids.
Oh.
So we've got sort of this blended family, you might say.
Two nuclear family,
Yeah,
sequential.
yeah. Yeah, I always tell people that marriage is not supposed to be a replicated experience. It sometimes is, but it's not supposed to be.
Before and after now retirement, what did you do for relaxation and entertainment?
Well, I actually loved what I did. And I still miss it somewhat, but the time was right. So, I was pretty much, I guess most people would say, a workaholic, but I always loved what I did. The thing that changed in the second marriage, as well as when the kids get grown, and all the kids were grown, my kids were grown by the time I was in my 50s. So the trick there was that you had a little more time. And I got to go to a lot of really interesting places. So sometimes I wouldn't take my wife, sometimes I would, but I always made a plan. If I was going to go someplace, don't go there, hit the meeting and go home. And I did that for a long time, hit the meeting and go home. So, I'd been to Peru twice and had never been to Machu Picchu.
Oh, wow.
Okay. Well, I had an opportunity to go back. So I did Machu Picchu. I was on the IRRI, which is the International Rice Research Institute's Board of Trustees, which was a lovely organization, part of the international centers. Feel very, very lucky. I worked on maize for my PhD my whole career in wheat. And then the third major cereal was rice, and I was on their Board of Trustees. And you get to go to the rice countries, which doesn't often overlap with the wheat countries. So we would start doing things that would be learning things. So like we had a meeting in Burundi, and I looked at it, and it so happened to fly to Burundi, you had to fly through Nairobi. So we said, if we're going to fly through Nairobi, I've always wanted to do a safari. So I had a friend from Uganda that actually booked it, because if you book in America, you get exorbitant prices. Yeah, if you book over there, it was oh gosh, it was a third of the price for the exact same safari. So we did a safari, and then so I've always loved traveling. And if you're in agriculture, it's like you get to go to places where a lot of people, they don't hold big meetings. It's like it's easy to go to Beijing for a meeting. But I went to Ulaanbaatar, Mongolia, and that was delightful.
Wow.
Okay. I've been to Khartoum, Sudan. I've been to Aleppo in Syria, when times were safe. But he did. So I always loved traveling. Okay. And so that's the main thing that I would do my work. I would help people, so if there's something like if someone said, we need a consultant. I consulted with ICARTA on a program review, just 2 trips to Syria both times, both when they were on the bad list. And most people don't know this, but the government of the United States has a very good policy, and I think it still works, though I'm not sure, that we will never use food as a weapon. So if you're in agriculture, you can go to anywhere in the world on an agricultural thing because we will never use starvation or food as a weapon. So we went to the Sudan when they were on the naughty list, and Syria when they were on the naughty list. You got to be careful. You got to do all the things properly. But the government, our government has always said food and starvation are not weapons. So we got to see things. Well, then when I retired, I got more time to do things. The things that happened, like I said, we would travel in January and February. We would travel a little bit in August for a short time. Okay. Now it's like, July, that kind of thing. March, I'm not crossing in the greenhouse.
That's what freedom is like.
You get 12 months out of your year. And I should mention, we talk a lot about students and the like. In a breeding program, I think you have to recognize how valuable your technologists are too. At the end of my career, I had two of them who were with me over 20 years each. They were magnificent. And before that, we worked with a ton of really good people. And the team that we worked with, they also had stability in the technical people. So they were really great. So Stephen Magula's technician was wonderful. Devin, and before that, Dave Shelton, before that, Paul Matern, their technologists were great. These were all people that really made the job easy, made us look really good.
All right, Stephen. Thank you for telling me your stories and all about your interesting life. And maybe we'll see you around.
Sounds good to me, Mark. Thanks for the interview. Really appreciate it.